regulatory standards that we have to abide with before you even use this radiopharmaceutical. Then you have to maintain institutional credibility. If there is any mistake in any of our radiopharmaceutical, they can shut the whole institution down. Because when you cannot joke with radioactive materials. Next slide, please. Now, if you remember, we made mention of some regulatory framework. Just as a reminder, you can talk of World Health, International Atomic Energy Agency guidelines, World Health Organization technical reports, United States Pharmacopoeia and European Pharmacopoeia standards, national nuclear regulatory authorities, and good manufacturing practice requirements. These are some of the regulatory agencies that ensure that whatever that we are using to abide by the rule, there is no mistake whatsoever. Because if there is any small mistake, you are endangering the life of what? The patient and the what? And the what? The clinicians. Next. What are key quality control parameters? You talk of radionuclide purity, radiochemical purity, chemical purity, excuse me, sterility testing, bacterial endotoxin testing, pH and visual inspection, filter integrity testing. Thank you. I cannot drink water, I'm fasting. So just bear with me. Now, radionuclide nucleic purity. We have to talk of the percentage total radioactivity due to intended radionuclide. Verified using gamma spectroscopy or half-life determination. You ensure absence of unwanted radioactive contaminants, and this is critical for technetium-99 and fluorine-18 preparations. Now, radiochemical purity. Fraction of radioactivity in the desired chemical form is very, very important. Then you measure this using TLC, paper chromatography, and HPLC. Then there must be low purity. Low purity may lead to altered phase
Hello, Mark. Can you hear us?
Now we can hear you. Yes. I'm sorry. Should I start my presentation?
Can you please display your slide?
Yes, but it's you were muted all the time, so I wasn't sure. Yeah, no. You.
Mr. John.
Can you hear me now?
Can you hear?
Yes. Can you hear me now?
Good morning.
Hello?
Hello, good morning, Mark. Can you hear me?
Can you hear me?
Yes.
Perfect. Thank you so much, guys. It was muted from our side for a long time, so yeah. I don't know where you are at the program, so I will just jump in with my presentation.
All right, now, please give us a minute. I'll get back to you in a minute.
Good.
All right, Mark, please, you can begin with your presentation.
Thank you. Can everybody hear me?
Yes.
Are we good?
Yes.
Perfect. Okay. So I was asked to talk about radiopharmaceutical research, and so today I was asked to cover the topic of the research protocol, so how you go about to make a novel radiopharmaceutical, how you should design your laboratory, and also some current trends in radioligand therapy. So as I am in the field of oncology, I will focus mostly on how to design a targeted radiopharmaceutical for cancer. And I hear that you already know what PET-CT is. So I will just say that the, sorry, I hear people speaking on the call. Can you mute, please? Okay, so you have the radionuclide. That is where you put the radioactivity. You just get a pointer. So here is the radionuclide that you put in a therapeutic or a diagnostic radionuclide. Then you have a vector, which can be a biological molecule, a small molecule, or maybe even something bigger like a nanoparticle. You have your biological target, which is the receptor or the enzyme or the disease process that you target. And then in this case, since we are working with oncology, you have your cancer cell. So your cancer cell express a certain target that you identify that you want to target. You design a molecule or a pharmaceutical that will target this biological target, and then you add your radioactivity to it. Then we select patients by doing a diagnostic scan where you can see where the cancer is, and you treat them thereafter with a therapeutic radionuclide, and then we hope to see such a good progression. So, for example, I have here the diagnostic application of fibroblast activating protein. And as you can see here, you would image the patient with a fluorine-18 fibroblast activating protein targeting a radiopharmaceutical. So what it means is we have a small molecule, and then fluorine-18 is bound into this molecule. This molecule targets what we call a receptor on the fibroblast, the activated fibroblast. So it's an immune cell that is activated and express certain receptors. And then in the end, these receptors or the fibroblasts are surrounding the tumor, and then it's going to sit around the tumor and image where it is, and then you can see on this patient scan that we have a tumor. So as an example, to put it further, we have fibroblast activating protein, which is a novel radiopharmaceutical, and it was the Society of Nuclear Medicine and Molecular Imaging Diagnostic Tracer of the Year in 2019. And here is all the types of cancers that can be diagnosed, so we can see the amount of tumor, also that it is present and that it expresses this certain target in many types of cancer because it is imaging the immune system. So the pipeline to develop a novel radiopharmaceutical we should understand before we go into how to design one. So first, and this is prostate-specific membrane antigen, which is a very well-known radiopharmaceutical in the field. So first you have the target. So in this case, it's the prostate-specific membrane antigen. In the previous example, it was fibroblast activating protein. So then you design a ligand. So we call it a ligand, but it's a molecule that will go to those certain receptors wherever they are expressed. And then you have the radiochemical optimization and validation where you radio label this vector that you have chosen, make sure it's pure. I think the previous lecture really nicely said about the quality control and all of that. We have to inject pure radiopharmaceuticals. Thereafter, you evaluate it in cells, so it's in cancer cells mostly. And then you do studies in animals to see if it's safe for human use and if you can actually see the tumors in biological systems. And then you go into clinical translation, where it first most often becomes a diagnostic tool to image cancer and diagnose it. And later it can become therapeutically applied to treat the cancer. So I'm going to use my own example just to guide you through the process today. So firstly, you have to decide what type of vector you want. And there is definitely different types of vectors. You can have small molecules, what we call affibodies, peptides, antibodies, full-sized antibodies, and then indeed nowadays we also use fragments. So when we identify the binding site of an antibody, we can pick a fragment thereof and make a smaller molecule just to look at the pharmacokinetics. So each of these molecules have a different profile pharmacokinetic-wise. So I normally work with peptides because I know they have a very short biological half-life in the body. So within 2 hours, they will be their target and then they can start doing their job and we can image the patients. Antibodies can take sometimes 3 to 4 days and small molecules can be very quick. Okay. Then you choose a disease to target. In my case, I am going to focus from now on colorectal cancer. It's the third most common cancer in the world. Prostate cancer and breast cancer is still higher up on the list, but I was thinking that this is quite a disease that has no therapy when the patients are reaching stage 4. And so it's actually one of the most deadly cancers in the world currently, with around 900,000 people dying every year from this cancer. So I thought this is a good target to choose because, of course, we need to have enough patients to do all the to actually say it's a good idea. So you have to have patience so that you get funding and so that people come on board for your development. If you choose a rare disease, there is no funding, and then you can, of course, not do the research you want. So in colorectal cancer, there is many receptors. There is some that is for targeting the making of new blood vessels towards the tumor, which is called angiogenesis. There is some that is just expressed on the tumor cell surface. One very well known is CEACAM5 or CMET or EGFR. You might have heard of these receptors. There is also some immune checkpoints. So that is when there is immune cells involved in the tumor microenvironment, other receptors that is expressed on colorectal cancer, and then also the tumor microenvironment. So by choosing colorectal cancer, it's now important to know which receptors is available or disease processes that we can target. So then this slide is a bit busy, and I did email my slides to the hosts of this event. So if you want a copy of the slides, you can just ask them to go on it in your own time. But basically, we need to know now how is this target overexpressed? Is it only in late stage cancer? Is it showing a sign of more aggression? Is it clinically relevant? So if the target is expressed, does it mean anything to the clinicians? Maybe overexpression of a certain target. So I can take an example. If you
This one. Sorry, ma. We are doing exams now, 8 to 11. So, but she couldn't continue.
is everything okay? Can I continue?
I want to retrieve for Alpha.
I'm sorry, what's wrong?
So sorry about that. Please continue.
Can I continue? Okay, because I hear a lot of background noise. It's not really fun to present like this. Sorry, guys. I know it's technical issues. Let's continue. So if you have a clinically relevant target, like say you have CEACAM5, we know CEACAM5 is overexpressed in colorectal cancer that is very aggressive. And so the clinicians would be very interested to have a diagnostic radiopharmaceutical, which can then show them which types of patients are very has to get a very quick treatment, is in late stage, and maybe has resistance towards current therapies. And in that case, also a therapeutic radiopharmaceutical will be of interest. Then once you have your target, you screen your peptide. And you choose one. You can use various sources of getting peptides. Normally, it's quite nice in literature. There's already a lot of peptides that targets these receptors that is being described in literature for non-radiopharmaceutical applications. So normally we go check in literature what peptides have been described. They are very nice and easy to manufacture. They are very selective, have a high affinity, smaller size. You would then add the necessary components that you need to radio label these peptides. We call it chelators. So it's a chemical part that accepts the radioactivity. And then you will check also if you can continue further. So just to show you that the selection of peptides, if you choose peptides that is already in the body, it is a very easy translation. If you go to other sources of peptide identifications. So say we take a computer, we call this in silico design, and the computer screens the crystal structure of the receptor for peptides. This is a medium risk, and yeah, it's quite nice if you can do this, because then you can identify peptides that has never been described before. Drug-derived peptides, this is peptides that have been already developed for pharmaceutical use, but has not been used in the radiopharmacy, has a low translation risk, because we know these are already safe, and then we can push them rapidly into the clinic. So these are normally the ones that I prefer. So then the chelator structure I talked about, it's important to choose the right chelator. If you think about the radioactivity in the future, you want to put into the chelator. So say you want to put a diagnostic one and you have a generator-based system because you don't have access to a cyclotron, you would normally choose gallium, and then you can use DOTA as a chelator. vector. I'm not going to go much into this, but please follow up with an email if you have questions. But just to say, the chemical design of the chelator that you link to the peptide is important if you want to choose certain radioactivity to work with and put into your radiopharmaceutical. Thereafter, you would do radio labeling optimization, and then here comes the quality control that the previous presenter talked about becomes very important. So you need to label with as little as possible of the vector, because you have a tumor there that has a certain amount of receptors, and you need these to be occupied by radioactive compounds and not cold compounds. So normally you have to spend a lot of time to make sure that you can label it is pure, it is a low amount of your vector in this mixture, and that you can have a very good pure product to inject and to test further. This is a very important step, and this can take the longest time for us of all the steps in the pipeline. We have to make sure quality control is okay. So you have to do cold labeling or labeling of a non-radioactive counterpart. So if you have gallium-68, which is radioactive, you also have to use natural unradioactive gallium to make the compound. And then you should characterize it with LC-MS and use that to actually validate your HPLC methods. So we need to know that our quality control methods are robust. They can definitely show us purity and identify our compounds before we go further. We need to know we made what we said we are making. Then one important factor that is often neglected is stability. And again, you need your quality control methods to prove stability, but we can have various different types of instability. It can be that your radionuclide doesn't fit very well into your radiopharmaceutical and it comes out. It can be that certain things in the blood takes it out of its radiopharmaceutical, or that when it decays, it also becomes unstable, but it can also be in the biology unstable here. So when you inject it in a live animal system or a human, it can be metabolized. So we need to check for all of these stabilities, because if the radionuclide is removed from the radiopharmaceutical goes to the target, but the radionuclide goes completely somewhere else, and then it doesn't give you what you needed. So then just to say the stability, and we talk about HPLC again, and here I just present the work of my student Sandile from South Africa, just to show you if you have labeled lutetium, PSMA, INT, for instance, you will see on the right-hand side a pure compound, and on the left-hand side, it's the same compound, same formulation or same batch that was evaluated around 2 days later, and then you can see a really big impurity forming. It's not free lutetium, but it's the peptide has been changed, and so it will not do its job. And if you inject this then into a patient, you will only have the activity that is in green there that will do its job, and the rest will be giving you other areas and go to areas you don't want in the body, for example, the kidneys or the liver, or places where it can cause damage. So that's also really important to do. Here is just an example of preclinical work. Firstly, the first HPLC chromatogram that I called end of synthesis was when we labeled it, put it on the bench, and after 120 minutes, you can see the second chromatogram on this. It is still quite pure. We actually incubated it with serum, and after 24 hours in serum, you will see that at around 8 to 10 minutes, there is some instability being formed. And in this case, that was, for us, not major concern, because as you can see, it's not really a big deal. And it's also after 24 hours in serum stability. But when we injected it into the mouse, you could see even 20 minutes after injection, there is really a big peak at 8 minutes. And so it shows you that what you test on the bench is normally, you should check in animals. And that's why we still do animal testing, because a biological system of an animal is a different story compared to that of, what you do on the bench or in test tubes. So You will see here the mice that I put on here has a lot of gut uptake, and we assume it's this instability that we see after 8 minutes. After all of this, when you know you have a stable compound, you can do some cell work to see if it has the same affinity and specificity. So does it bind to the receptors where we said we should bind to? You can also do a lot of more advanced cell culture work. 3D cultures is when you make small tumors yourself in a test tube and test it. But yeah, for the moment, we normally just do basic two-dimensional cultures, which is the first one. So we see some cancer cells that comes from patients, and then we test if our radiopharmaceutical binds to these tissues, and also if we have some therapeutic activity. Finally, we have the in vivo analysis as well. And in this case, we check pharmacokinetics. So we check how long does it stay in the animal? Does it get excreted? Does it go to the tumor? And in this case, you can see we have a very high tumor uptake. If you look at the second graph, you see there is positive tumor. It's the biggest bar, and it's very high compared to the kidneys and the liver. So this was an excellent radiopharmaceutical in the mouse system. You can see there is no background. So if you compare this mouse to the one I showed you before, you would agree with me that the one on this slide has a really high background and uptake in this gut area, so it doesn't look so great, although there is a high accumulation in the tumor still, whereas in this one you just see the tumor and then the excretion in the bladder. So yeah, it's being excreted quite fast, so it removes all the background, and there is a high accumulation in a positive tumor. So normally this is for us very great to see, and then we suspect that it would be good to take this radiopharmaceutical into humans. You also have a wide variety of animal models that you can choose. So you can make cancer in the mice by giving them some toxicity, or you can make mice that are engineered to express some tumors. You can also take human cells, what we call the cell-derived xenograft models, where you take human cells and you inject it into the mice and it grows human tumors. This is the one we use most often and is the easiest to use. And then you get patient-derived xenografts, where you actually take a patient tumor and plant it into the mice, and then you grow them. But this is a more complex method. So we normally just inject tumor cell lines that we have into the mice and evaluate if the radiopharmaceutical goes to the target or not. After that, you can do therapeutic efficacy, where you have all of these mice that grow the tumors, and you give them the therapeutic radionuclide and see if you can increase their survival and stop the tumor growth. And then you can also look at safety and toxicity and do histopathology and conventional toxicity studies that you would do for other radiopharmaceuticals. And then hopefully by that time, we are ready for translation into humans. I think very importantly, before you go into humans, is to make sure you have a good GMP compliant production. That's when we make sure our quality control methods and radiochemical purity is of good quality, and that we can exactly tell people what we inject into them. You have to have all of your mouse data and your cell culture data ready. You also have to maybe focus first on the diagnostic, because therapeutic radiopharmaceuticals is difficult to translate. They can be toxic. You need industry to also buy into this because it's very expensive to do. But normally you would make sure, like the previous lecturer said, to make sure that everything is very pure and safe and sterile before you inject it into humans. So that is how I would say is the whole pipeline of radiopharmaceutical development. I know I gave you a lot of concepts. Unfortunately, today we don't have that much time to talk about everything in detail, but my slides is available, and you can definitely get a copy thereof from the host of the organization of today. So I will actually just go into quick one slide of the design of a radiopharmaceutical research laboratory, and then thereafter we will talk about the novel trends in radiopharmacy, and then I will stop there. So The design of a radiopharmaceutical laboratory can be very expensive, so I would highly recommend that you have some of this in-house, but also get collaborators from other areas to provide you some of these services. So organic chemistry and chemistry, maybe peptide synthesis. This is to generate the radiopharmaceuticals themselves that you will start to label. In this case, you can either work with a chemistry department or you can buy these ligands commercially. So you can design them and send them to somebody. We use GL Biochem in China, and they make it for us and ship it back in good quality, and then we can continue. But you can also manufacture them in-house. But remember, if we say organic chemistry setup, it's a whole department. So if you have to put it into your own research facility, you at least have to have a decent organic chemistry lab. And there is a lot of analytical equipment like NMR and these things that you need access to. So it can be quite expensive. The second laboratory that you have to have access to is a radiochemistry laboratory. It has to have basic labeling equipment like a heating block, a fume hood. It should have adequate shielding, and you should plan ahead for what radionuclides you will work with, because each radionuclide requires different shielding. You also have to have other equipment that you might need, like centrifuges, based on your labeling methods, nitrogen, all of those things. Then you need the production of radionuclides. So in this case, I think this is also an expensive output. So either a cyclotron on site or access to generators that you can use, the gallium technetium generators, or maybe even rhenium, or through collaboration, maybe with areas where they produce in reactors, lutetium, and so on. Then you need access to a radioanalytical laboratory. And for preclinical and research and development, this is a lot more intensive than what you need in the clinic. So you need LCMS, which normally we don't use in the clinic, ITLC, radio HPLC, gas chromatography, and maybe even a high purity germanium detector. So there is a lot more of equipment in your radioanalytical laboratory that you need for preclinical and research and development compared to your clinic. Finally, a basic cell culture laboratory is definitely needed, gamma counter, and more specialized equipment. You need a fume hood, not a fume hood, you need a biological safety cabinet, storage for your cells under nitrogen, microscope, incubator, centrifuge, all of these things. Again, you can work with another department that already does cell culture, but then they should be okay with you working with radioactive compounds in their cell culture laboratory. And this is quite an important step in our development pipeline. So my opinion is maybe make sure you have a good radiochemistry laboratory, a radioanalytical laboratory, and a cell culture laboratory, and then you can collaborate on the others. So I would collaborate on the production of radionuclides, the organic chemistry, and the animal studies, because this is a lot of infrastructure needed for one institution to carry. The animal housing and imaging equipment is definitely something you can outsource or collaborate with some other institution. I know many people want to have these themselves, but it is equipment intensive and a heavy cost to carry if it's only for your own use. To use an animal facility that has radiopharmaceutical capabilities intensively, you need to have many researchers on site to do research, because an animal study takes about 1 week, 2 weeks maybe. So if you think of housing an animal facility for 12 months, 4 weeks, then you need a lot of research to go through that place to make it worth the money. So I would often say, if you are starting out, you should collaborate with other institutions. So animal housing, you need the cages, you need the animal housing that is according to veterinary standards. You need a lot of personnel. You need SPECT and PET cameras and all of that. So it can become quite intensive. Okay, so that is what I wanted to say about the design of a new radiopharmacy development laboratory, just some small current trends in radiopharmaceutical research to end off. So currently we are not, you would be very familiar with beta minus emitters that is in the middle. So that is lutetium-177. We have used this always for therapy. The current trend is to use alpha emitters like actinium-225, astatine-211, and lead-212 for therapy. And also Auger and internal conversion electrons are gaining interest. Now, this is for research purposes and not yet for the clinic. The clinical implementation of alpha emitters is slowly coming along. Auger is not yet into the clinic, but alpha emitters does a very strong DNA double-strand breakage, so it's even more powerful than the therapy we have used in the past. So if your patient is resistant to beta minus therapy, alpha emitters can be very attractive. Auger emitters are very small ranges, so they actually just target single cells with very high energy. So it looks attractive, but it's very complicated to implement, and a lot of research is being done on that. Second new trend is to use the terbium radionuclides, especially terbium-161. I think you also in Nigeria have started with it or had the offer to work with it. But there is, for me, the idea of terbium-161 was not to be better than lutetium. Somebody somewhere thought it would be interesting to use all the terbium radioisotopes. So we have terbium-149 as alpha emitter, terbium-161 as Auger emitter, terbium-155 as beta minus, terbium-161 as beta minus and Auger, terbium-149 giving you PET. So it was quite interesting for everyone to say we make one molecule and then we just put in what isotope we want to do what we want. But this is in practice not even close to a possibility at the moment. So only terbium-161 is used on a wide scale. And for example, terbium-149 cannot even be produced at the moment in an amount that is big enough to treat humans. So again, purely research, not really clinically practicable at the moment. One of the future perspectives that is going into the clinic quite fast is to use antagonists rather than agonists. So you have dotatate, TOC, and NOC, which is a somatostatin receptor agonist. The problem with agonist molecules is that it gets internalized into the cell and then it does have some effect and it's inside the cell. So normally we were quite okay with this because then your radioactivity is shielded and cannot get out of the cell. Nowadays, they say, let's start to do antagonists that just localizes on the cell membrane and does not get internalized. And they see that these work better because they have a higher affinity for the receptors. So does it work? I have no idea, but you might hear that dotatate can be replaced by JR11, or the other molecule is LM3. So this is just some of the research that is actually already going to the clinic. And when we design novel radiopharmaceuticals, we also now have to consider, do we use receptor agonists or receptor antagonists? Clinical research that is going forward is the fibroblast activating protein that I talked about earlier, where people say it's comparable to the use of FTG. It's being developed for technetium and gallium radiopharmaceuticals as well, so it's definitely still relevant and coming to the clinic. There is also a great movement to develop radiopharmaceuticals that doesn't just target one single cancer, like what I said, the colorectal cancer in my case, but maybe like the fibroblast activating protein, like I showed you way earlier, something that can do this, target many types of cancers. So this is when you target the tumor microenvironment. The tumor microenvironment is everything that surrounds the tumor. Okay, I just made out everything that surrounds the tumor and not really, I hope you can see now. Let's just open it up again. Stop share. Everything
ma'am.
that's yeah, just hang on a second. Don't worry. I just want to make it nice. Everything that surrounds the tumor and not necessarily a cancer cell itself. So that is what the drive is to do the tumor microenvironment targeting. And as you can see, there is the fibroblasts, like I said, FOPI, but there is also the tumors that develop hypoxia. So if a tumor grows, it has areas where there is no oxygen anymore because there is not good blood flow, and you can target that. You can target the acidity of the tumor, so maybe make some radiopharmaceuticals that go accumulate in areas of low pH. The increased metabolism, as we know, FTG has been done for a long time, and this is also typically because you can use it for all types of cancers. So yeah, some people say, why are we targeting a single type of cancer and a single receptor when we can target one type of radiopharmaceutical for multiple cancers? And I think this is the last one, maybe. Finally, also the pre-targeting of antibodies. Since I told you before, antibodies takes around 2 days to circulate. Some people say we should inject the antibody alone, let it circulate for the 2 days, and afterwards inject a radiopharmaceutical molecule that will go target the antibody inside the body. So you first let the antibody go to the tumor, and afterwards you inject the radioactivity, and it goes stick onto those antibodies that is already in the tumor, and then it's just a short time the patient needs to be in the radioactive area. So this is also an area of research. I just want to thank my lab, because always I do. They are my constant support. And I thank you guys for listening. I don't know if we have time for questions, but this is what I wanted to tell you today. My apologies that I went so fast, but my lecture is available afterwards. Thank you.
Thank you very much. Let us appreciate Dr. Jenka. Yeah, any question, either online or here in the hall? Any question, either online or we in the hall here? If you have any question, please signify. And if you want to write it out so we can read it to her. Any question? Okay, there's no question here. Online, any question online?
I will put my email in the chat, and then if anybody has a question, they can email me afterwards, or if they want a copy of the presentation, they're welcome. Okay.
Yeah, since there's no question, yeah, let's appreciate her again, ladies and gentlemen. Let us clap for her. And
Thank you.
the organizer of this program have promised that the slides for the 2 lectures presented today will be available. And also, I need to apologize that until this morning, we never knew there's going to be a strike in UCH, and that is what causes this little issues, the space and other technical issues. So please. Forgive us, is because of the strike. Now the molecular laboratory. It takes like 10 people at home. So we will use the attendance to pop up the 10 people that will be visiting the place.
Or just wait.
David.
Yes, sir.
Yes, sir.
Are we done for today? Is it she and